All right, hi everybody, and welcome to the Center Seminar Series today. Before we get started, I'd like to acknowledge that this meeting is being hosted on the traditional lands of the Bindal people and pay my respect to their elders past, present, and emerging. Our expectation is that the interactions during seminars are constructive and that all attendees behave with respect and consideration for others. So before we start, just a bit of housekeeping. If you could all please mute your microphones, which I can see you all have. And then I'll introduce Renato. And then following the talk, we'll have a bit of time for questions. And you can either ask your questions directly or put them in the chat box and I'll coordinate that. So today I'm super excited to be introducing Dr. Renato Morais, who is a coral reef ecologist here at JCU. I first met him at the end of our PhDs. We were both finishing around the same time. And since then, we've actually been collaborating quite a bit together on a number of projects, which has been really exciting. So Renato's work for his PhD, was focusing on developing a new metric in coral reef ecology that goes beyond some of the more traditional indicators that we typically used to assess things like reef health and ecological function. This new metric is called potential fisheries productivity. And what it does is it moves beyond quantifying things like biomass, which is what we normally measure on coral reefs when we're looking at the impact of management or we're trying to assess fisheries status, things like that. And this instead incorporates time into that. And that actually has a bunch of quite profound ramifications, which he's been working on for the last few years. So he's been focusing on mostly ecology questions to do with reef fish productivity. But then we began, sorry, it's a fly in the room. We've begun collaborating on some of the fisheries questions that entails as well, because there's a whole bunch of things that suddenly this metric, which is really simple and quite elegant, can suddenly address, like which fish are the best to catch, which are the most sustainable. Why do we get some fisheries in heavily exploited places that still seem to support people's livelihoods? So there's a bunch of questions like that are quite profound that this new metric has been making heaps of progress towards. And also, he has just been accepted for the prestigious Bronco Weiss Fellowship, which essentially gives him 5 years starting next year to go do his own research project on almost anything he wants, it sounds like, which has a startling success rate of 1.6%. So I think we can all give you a big congratulations for that. And without further ado, Renato Morais, go ahead.
Well, thanks for the intro, Pat. Thank you very much. I don't think I even need to continue with the seminar because you presented everything I'm going to say today, but much more elegantly than I can. So I'll just ask everybody to forget you heard what Pat said and pretend I'm going to be telling you something you don't know, okay? I'm going to share my screen now. No, thanks for that, Pat. That was awesome. And okay, so as Pat said, you probably are seeing a lot of people. I'm gonna just put myself here. Okay, so as Pat said, I've been developing in my PhD this metric. I wouldn't say it's new. The idea has been around for quite a long time. It's just that I formalized it in a way that may be easier to execute for coral reef ecologists. So I'm going to be talking about the principles behind this and a little bit of the work I've done in my PhD, which was developing the case for it and then presenting some ecological examples. I'm going to take Patrick's photo from here. Okay, so I'm not going to be talking anything of what I've been doing in the postdoc, which involves the collaborations with Pat at this point. So this is going to be focused on stuff I did previously. So the background of the talk is, as we all know, reef fisheries are an important source of food and livelihoods for people, particularly on low-income countries. Again, as we all know only too well, many tropical reef ecosystem or tropical ecosystems, and particularly coral reefs, are suffering disproportional effects of climate change, and reefs are suffering widespread loss of corals from bleaching, severe coral mortality coming from that. So the obvious question that arises is, can these tropical reefs, can these coral reefs now threatened by climate change continue to provide the resources that people need to extract from them? So obviously, I won't answer this question, and I won't even ask this question in this talk, but I would like to talk a little bit about reef resources, and particularly reef fish resources, and how also I believe we can do a better job in quantifying resource production on coral reefs. So a key term underlying this talk and my work as a whole is, as Patrick said, biomass production or productivity. Animals produce biomass when they grow. When they go, they incorporate energy and nutrients in their bodies, which they source from their food. Production of new biomass then supports key or important ecosystem functions such as predation, but also important ecosystem services, such as, for example, the provision of fisheries resources. But why not talking only about standing stock biomass? Why talking about biomass production? What can it tell us that's different? So Pat sort of hinted at it, but the thing is that biomass alone doesn't tell you anything about the time frame in which it was accumulated, right? So it's a bit like your savings account. So if your savings account is big, it could be that you're a very cheap person that saves a lot. It could also be that, those are your lifetime savings. We wouldn't know. But biomass production is time explicit. We know exactly to what time frame we're referring to. So it's more like your fortnightly or your monthly salary. You know exactly when it's going to come. And this contrast between these two, which are obviously not the same, is particularly important for reef fishes because species, they may vary a lot in their longevity. So if your biomass savings account is filled with cyan gobies, and these cyan gobies live for a maximum of 59 days in the wild, that will be a very different situation to if your biomass savings account is filled with midnight snapper, which can live for up to 81 years. Now, that's obviously an extreme scenario, and nobody would want their fisheries savings account to be filled with cyan gobies. Nobody would want to eat gobies. But even when we take into consideration the same species, there may be huge variations in lifespan among individuals, right? So pick up the line surgeonfish on the Great Barrier Reef, for example. An individual that's 18 centimeters long can be as old as 5 years or as old as 45 years, okay? So when I'm talking about biomass production, in essence, I'll be talking about a unit of mass or weight, as you prefer, of fish, relative to a unit of area, relative to a unit of time. And I will use the words production and productivity interchangeably here, because although they are distinct concepts, one is the area under a curve and the other is the tangent of a curve. If we consider a short enough time period, they should approximate each other and be approximately the same. Okay, so if we think of the general life cycle of a reef fish, which is sort of split between a coral reef and the open ocean, an individual fish that's on the reef will grow until it becomes an adult, and then it will start reproducing, it will start spawning. That's sperms and eggs can be broadcast directly in the water column, or they can be attached to the reef substrate. But in any case, the larvae that eclode from these fecundated eggs will become larvae in the open ocean. And the few ones that survive will then develop into free swimming larvae that are then ready for the next step, which is recruiting back to the reef. Okay, so from the steps that are relevant to the reef habitat directly, spawning means that there's a net loss of biomass for the community. Recruitment means that there's a net gain of biomass for the community, but that gain is likely to be small because recruits in general are very, very small in size. although recruitment is very important for other reasons, as it's what actually drives population persistence. But if we think exclusively from the optics of biomass produced, so the vast majority of biomass produced on a reef will come from the growth of individuals. So if we look at the proportional distribution of biomass between these different processes at the community, and here we have the different colors hinting at that, we can see that a large part of the biomass cannot be harvested. That's everything in the top part of the donut. So that includes biomass lost to mortality and through reproduction, but also biomass that's locked into newly settled recruits and individuals that are still not big enough to be fished. So the biomass that's actually harvestable will be a proportion of the previous biomass and the newly produced biomass from new growth. And then if we further divide that, we can imagine that the sustainable harvestable proportion of those resources is what was produced via new growth. Because we obviously only include the individuals that have already grown enough to be harvested. Okay, and how can we quantify fish production or productivity? Well, we can use the simple 5 steps of individual age modeling, and I don't want to seem like I'm selling you anything, but these simple 5 steps, they start where most reef ecologists start, right? With field data. So the most common method of getting field data for reef fish is underwater visual service. So in this method, we generate species and most importantly, individual level data. And this individual level data includes species identities, abundance counts, and some type of estimate of body length of the individuals that have been surveyed. So that takes us to step 2, which is we add on top of the field data based on species identities, relevant life history trait data that can carry us through to the next steps. So in this case, where I'm talking about length weight conversion factors on growth curves and mortality trajectories. So we use the length weight parameters to convert fish lengths from the surveys to body mass. And when we multiply individual body mass by species abundances, then we obtain the classic standing stock biomass. So that's what we do. That's what we've been doing for 70 years. That's the norm. So, but things don't stop on step 3. Then on step 4, what we can do is we can use the species specific or the population specific growth curves to actually estimate the somatic growth of the individuals we surveyed. And how do we do that? Well, we will age those fishes from the surveys. Bear with me a little bit. I'm going to try to explain what I mean by that. So if you have your fish that was surveyed and you have a growth trajectory, which sort of reflects the way that fish is expected to grow, it's based on a growth curve for its species, then what we can do is we can position that fish in the growth curve based on its body size, and we can determine the operational age of that fish. Now, the operational age is not the same as the real age of the fish, right? Because we know that real age varies a lot and can vary a lot of fish that are the same size. But the operational age is the age relative to the moment the fish stops growing. That's what matters for us at this point. So 2 fish from the same population that have very different ages, which we don't know, will have almost exact same, the almost identical operational ages if they both are close to their asymptotic size. Anyway, back to our fish from earlier. If we then have the operational age of this fish at time T1, which is when we counted the fish, and its growth trajectory, we can then look at any posterior time, let's say T2, and then estimate the size, the likely size of that fish in T2. And from that, we can calculate the expected increment in size between T1 and T2, and that's your growth. So that takes us to step 5, which is incorporating natural mortality by using mortality rates to determine the likely losses due to natural mortality. Now, estimating growth is quite simple, but predicting mortality, it's impossible. And what I mean by that is that we can never tell at which age or when a fish will stop being alive and start being dead, okay? But we know two things that can help. First, we know that fish tend to settle in cohorts that are abundant, and only a few individuals actually make into adulthood. So they suffer extensive mortality along the way. And the rate at which these cohorts get smaller and smaller every year as they age decreases exponentially. And we also know that the mortality risk an individual fish experiences through its life decreases exponentially as body size increases, because small fish literally fit in many more mouths than large fish. So they have a higher predation risk. So if we overlay these probabilities, we can then take a probabilistic approach to the fish being alive or dead. So it goes like that. We can, for example, we have our T1, we can pick another time T2, and then we can simulate stochastically whether the fish is going to be alive or dead. So if the fish survives, its growth will count towards our final metric of productivity, otherwise it won't. Okay, so those are the 5 steps to productivity. Now, that brings a question, which is, do we have access to life history data that can actually allow us to estimate growth and mortality? Well, I would say that in an ideal world, you would get age estimates from otoliths. Otoliths are these ear bones on fish that, if you section them, they have rings that are deposited annually. So ideally, you would have a sample of individuals from all populations from your community. You would get otolith samples. You would then get size at age data from which you can then reconstruct both growth trajectory and mortality trajectories based on something that's called the catch curve. But obviously, this is not feasible most of the times. You will have hundreds of species, and you will normally be working at multiple geographic sites, and sometimes even, the whole globe is your universe. So what we can do instead? Well, the alternative is to try and model a specific parameter that can describe these models based on data that we can compile from the literature and some informative type of predictors. So I won't get into details on this, but if you are interested in this framework as a whole, then I have described the principles behind this method, including those 5 steps in that paper on coral reefs. And I also have an R package, which is called rfishprod. It's ready to use on GitHub. And with rfishprod, you can use the equations to estimate productivity, but you also have predictive models for both growth and mortality trajectories for reef fishes. And the address to download, if you want, is just up on the screen. Okay, so now I'd like to present some case studies so that I can, hint at what type of ecological insights we can gain when we use biomass production, not only as a metrics of fish and also, as Patrick said, potential fisheries resources and production, but also as a measure of ecosystem function. So case study 1 is a study in which we wanted to understand what's the relative importance of different energetic pathways in determining fish productivity. And if our perception of the importance of these pathways would change if we, switched between biomass to productivity. So an energetic pathway or a trophic pathway is a group of trophic interactions that lead from one type of resource to a consumer, okay? And we define 5 main trophic pathways through which fish would obtain their energy and nutrients on our coral reef. That's the name of those. We have a zooplankton pathway, we have an off-reef sand pathway, an epibenthic and cryptobenthic reef pathways, and we have another pathway which is just generalized and opportunistic predation. And we further subdivided zooplankton pathway into two components, zooplankton that's captured off the reef, normally by larger fishes, and zooplankton that's captured on the reef, normally by smaller fishes. So to capture the maximum of the fish assemblage we could, we combined different types of fish counts of different areas targeting different sizes and behavioral classes of fish, and clove oil stations, which are the best way for serving small and cryptobenthic fishes. And we did this study at Lizard Island on the northern Great Barrier Reef and across a habitat gradient from the exposed fore reef slope through the reef crest, over the reef flat, and down to the back reefs of the lagoon. We used two indicators, again, standing stock biomass and productivity or biomass production. And when considering biomass first, if we look, so each color here is gonna show one of the trophic pathways. We have the habitat gradient on the x-axis, and we have biomass on the y-axis. And we found that the two most important pathways were the off-reef zooplankton one and the epibenthic pathway, okay? With some habitat variation, like the off-reef planktivores were concentrated on the fore-reef slope and crest, and barely anything on the flat and the lagoon, while all the other 4 trophic pathways had a minor contribution to fish biomass. Okay, and when we looked at productivity, we saw that the 2 largest trophic pathways from biomass also were very important here, with some similar patterns. We also saw that, you know, those 3 smaller components were still smaller components to biomass, but a little bit more important than when considering biomass, particularly for cryptobenthic fishes. But the most shocking thing here was realizing the difference it made into our perception of the importance of reef planktivores when compared to biomass. So while their biomass was very small, their productivity was very high. So this is a group of reef fishes that we wouldn't really have perceived their importance had we only analyzed their biomass. So overall, averaging across the habitats, we had fish productivities by the different pathways at about 5 to 14% of these minor pathways, while for the epibenthic pathway, we had, you know, it composed 29% of total productivity, and the zooplankton pathway, which includes both the off-reef and the reef pathway, comprised more than 40% of total productivity. So to put this in another way, if you think for every kilogram of fish produced every day on that reef, more than 40% relies directly on food from outside the system. I thought that was pretty cool. And well, the second case study was a bit of an extension of the first one, except that here we weren't as much concerned with the different trophic pathways as much as we were actually concerned about understanding what was the impact of severe coral mortality on fish biomass and productivity and other ecosystem functioning metrics. Many of you will know that Lizard Island, again, same place I did the study from the previous case study, it was hit by 2 cyclones, one category 5 in 2014 and another one, a category 4, 1 year later in 2015. And then it was hit by 2 massive bleaching events that affected the Great Barrier Reef as a whole in 2016 and 2017. So it was a very dynamic, let's say, couple of years for the reef inhabitants at Lizard Island. So considering those same reef zones I mentioned in the previous study, if we compare the hard live coral cover between 2004, which was before all these events, and 2018, which was immediately after these events, what we see is that there was a sharp decrease in coral cover on the exposed habitats, on the slope and the crest, which were the habitats where coral cover was relatively high back in the day, while where the coral cover was already low, it just remained low overall. But at the same time, what we saw was that there was a reef-wide increase in algal turfs. So that happened across all habitats, but that was particularly important on the slope and the crest. So we wanted to know how fish productivity would respond to these important ecosystem changes. And so we compared with data collected for this study 15 years before. So for this study, we used different metrics of ecosystem functioning. Besides standing stock biomass and productivity, we also calculated something we called consumed biomass, which is the fish biomass expected to be lost to predation. And we also calculated turnover, which is a measure of how fast biomass is replaced by new biomass. So both of these, alongside productivity, they relied on being able to use that individual age framework I presented earlier to be actually estimated. So we found that in this 15-year period, there was actually a big increase in standing stock biomass on that reef. There was also an increase in biomass production and consumed biomass, although these were a little bit smaller. And interestingly, there was a decrease or a smaller decrease in the turnover of that biomass. So although that fish assemblage appeared to have become more productive, because turnover is a measure of replacement of biomass by new biomass, it suggests that replacement rates were just not as high in 2018 compared to before in 2004. So we then looked at how biomass on the y-axis again is distributed or was distributed across body sizes on the x-axis for the two time periods, 2003 in orange and 2018 in green. And what we found is that relatively small and medium-sized fishes appear to have increased in their representativity. And that was the same for the other indicators. I won't detail here. We then looked at which fish families could be leading this pattern. And what we found is that those changes were mainly due to three groups of herbivorous fishes: surgeon fishes here, parrotfishes, and rabbit fishes. And when we looked at their size structure, that is their abundance by size class for these three families, what we saw is that individuals with the same sizes that were driving those increases in biomass productivity and consumed biomass earlier, they had increased in abundance at the same time. So what this suggests is that those fish, they got larger between 2003 and 2018. Now, we think that the increased biomass and productivity of the system was being driven by herbivores responding to increases in their favorite feeding microhabitats, some sort of resource tracking, and their favorite feeding microhabitat, as we know, is algal turfs. Now, this has been shown to happen in different places, but most importantly, because these increases in productivity appeared to be due to larger individuals of herbivorous fish families, and because these many of these species of herbivorous fishes, they tend to live very long lives, so many decades often. So it's very possible that the fishes that have grown bigger and had produced more biomass recently are potentially the same ones that were there before. And so what this sort of hints at is that we may be looking at storage effects of individuals that were in the population and not real increases in productivity. So it's not that the system is responding to coral cover by increasing its rate of turnover, as we saw turnover decreased. So this suggests that this extra biomass may not be stable in the long run. So we need to interpret this with caution. Just a quick sip of water. So Finally, the third case study and last. So we wanted to evaluate how standing stock biomass and productivity are related across larger spatial scales. So how or if, to be fair, and also if fishing affects those relationships. So there's this general belief, unspoken belief, that fish productivity is proportional to fish biomass on coral reefs. Now, this is what I would call equivalence between the two. And this is implicitly assumed whenever somebody draws inferences on the productivity of reef fishes or reef fisheries by analyzing exclusively standing stock biomass. Now, this is a hypothesis, but it turns out that it's equally likely that non-equivalence is a thing as well. So non-equivalence can emerge from different types of relationships happening here, mediated by factors we don't know, or by a lack of relationship at all. So far this, which seems like a very simple question, hasn't been addressed on coral reefs, maybe because we didn't have a method to easily estimate biomass and productivity from the same type of data. So for this study, we focused on parrotfishes, not just because they're cool, but also because they are some of the main targets of reef fisheries pretty much everywhere in the tropics. And we used this large-scale data set of parrotfish counts, which included surveys from the Eastern Indian Ocean all the way to the Central Pacific. And what we found is that the geographic patterns of how biomass was distributed did not mirror the geographic patterns of how productivity was distributed. So some of the localities with the highest productivity, they had some of the lowest biomass, and some of the localities with the highest biomass had the lowest, some of the lowest productivities. So as a consequence, when we plotted productivity on the y-axis against biomass on the x-axis, using regions as our data points, we did not find a relationship at all. When we look at this same relationship, but at the local scale, so here each dot is a survey, then we found that there were actually different relationships going on. And these relationships, they could be divided in two sets. So the first one happening for high biomass regions. Here, the increases in productivity as biomass increased across surveys were relatively weak. And then we had the low biomass regions where the increases in productivity with increases in biomass across surveys were much steeper. And we also found that these high biomass regions, they had in general low human population densities, they had larger fishes in general, and those fishes had a lower turnover, that is a lower productivity per unit of biomass. And in contrast, the low biomass regions had larger human population densities, lower sized fishes, and higher turnover or higher productivity per unit biomass. So hence the steeper slopes in the previous plot. So this suggested to us that maybe it was human impacts and particularly fishing or overfishing that were actually driving these differences in how these relationships were happening. So what we did, we used a model to simulate fishing in model fish communities. So the fishing we simulated was what we call size biased fishing or size selective fishing. And so it happens when fishers tend to favor larger fishes and tend to ignore small ones. And then there's limits to what that they can capture as well. So this is a very common type of fishing on coral reefs. So what did we see when we simulate fishing on these model reef fish communities? Well, we saw that when we increased the exploitation rate, so that's basically a proxy for fishing, when we fished harder, biomass declined promptly, okay? But as we fished harder, productivity declined more gradually. It may be harder to see here. So let's pick a common reference point. So 0.75 exploitation rate. We see that there's only 25% of biomass left. That's how exploitation rate is actually defined, 1 minus biomass lost. But 75% of exploitation was still leaving for us about 50% of the original productivity. So it turns out that when we plot the two trajectories together, we see what we call these decoupled declines in biomass and productivity as you increase fishing. And they define this yellow area here, this yellow buffering area, which we call buffering productivity, which is basically a type of compensatory productivity. And what it means is, as you start fishing, the community starts producing more biomass per unit of biomass. So there's less biomass there, but it's producing more biomass per unit. So it's a little bit of a measure of resilience of the system, okay? So this type of compensatory productivity, this buffering productivity, may even help us to explain, for example, how sustained fisheries yields can coexist with depleted biomass sometimes on coral reefs. So going back to the hypothesis earlier, I think we showed that there's not equivalence between reef fish biomass and productivity. What we actually have is multiple different relationships that happen at the local scale and that are mediated by fishing. So with that, I'd like to finish by pointing out that I think we really need to start embracing what I've been calling a new resource assessment paradigm on coral reefs. We need to think more about process-based metrics, about dynamics, and we need to take into consideration how fast new resource is produced, how fast fish biomass is produced, rather than only assessing how much biomass is available at any given point in time. So I hope that my talk today sort of highlighted some of these issues, but also highlighted the opportunities we have now. It's pretty easy doing this now. So if I can help, please let me know. And if you're interested, please go after our fish prod. It's going to be a pleasure having people using it. And thank you very much for your attention. I'm happy to take questions.